everyone. I'm Richard Grewelle. I'm a fifth-year PhD candidate in the Department of Biology at Stanford University, working under the direction of Professor Julia DeLeo. I grew up in Kentucky, graduated from the University of Kentucky with degrees in biology, chemistry, and mathematics. Since my college years, I've worked very closely with human and wildlife populations at high risk of disease, as genetics to inform epidemiology and conservation efforts. One of the largest efforts in my PhD has been the investigation of a parasite known as the thorny-headed worm that is implicated in up to 25% of sea otter mortality here in California. I also work extensively in developing new approaches to model and treat schistosomiasis, which is a human disease that affects 200 million people globally each year and is second only to malaria in its impact on human health among the tropical diseases. The disease manifests as a complex suite of symptoms stemming primarily from the inflammatory processes of the body mounts in response to parasite eggs embedded in tissue. Despite centuries of effort to eradicate the disease, we have only achieved limited success. Current drug treatments aim to eradicate schistosomes in humans, but because snails also harbor a stage of the parasite, humans can be reinfected after re-entering water bodies following treatment. Successful elimination requires treatment of both snails and humans. Recent advance in genetic technology has paved new avenues to control disease through CRISPR-Cas9 mediated gene drive. Gene drive allows genes of interest to be inherited at a faster rate than usual. Genetic immunity to schistosome infection in a snail population can spread in this way. Lower infection in snails means less transmission to humans. I developed a series of integrated genetic and epidemiological models to investigate the conditions in which gene drive immunity could successfully reduce schistosomiasis. Importantly, I find that snail species with low to moderate rates of self-fertilization in isolated water bodies where disease risk is high are best targets for this technology. Proper design of gene drive genetic construct is also crucial to minimize fitness costs to the snails and prevent the rise of mutations that resist the spread of gene drive immunity. When this technology is paired with coincident yearly drug treatment, elimination of the disease happens more rapidly than with either alone, and without the gene drive immunity, drug treatment can reduce but not eradicate the disease. Modeling like this is crucial to understand the feasibility of implementing a new technology like gene drive, particularly in a natural system. It is wise to progress from in silico to in vitro to in vivo, with each step informing the next, because although this technology represents a new frontier for controlling disease, pests, or invasive species, the spread of a designed gene in natural setting carries ethical and practical implications. This model is an advancement towards a biologically realistic simulation and can serve as a template for future work in gene drive feasibility analyses.